This is the cyclic AMP pathway or G stimulatory pathway. Uh, in this pathway, we will study that how G proteins, G stimulatory, they are activated. So G stimulatory receptors, they are also called the serpentine receptors, as it looks like a snake, and it is also called 7-pass receptor or G protein coupled receptor. So this receptor, it 7 times crosses the cell membrane. First, ligand will bind to the extracellular domain of the G protein receptor, and that's why ligand, it is called first messenger. So once ligand is will bind to the extracellular domain, the signals are passed to the intracellular domain. This is the intracellular domain. So once signals are passed to the intracellular domain, the G protein initially, which is in inactive form, it is converted into its active form. Inactive forms of the G protein, it has alpha stimulatory, beta and gamma stimulatory fused with the GDP. Once it is converted to active form, the beta and gamma components are separated and GDP is separated. Alpha stimulatory will bind with the GTP. This is the active form of G protein. So this active form of the G protein, it will bind to the membranous enzyme, which is adenylyl cyclase. Adenylyl cyclase, so it will cause phosphorylation of the adenylyl cyclase, so that this enzyme will be activated. Once it is activated, it will cleave ATP into cyclic AMP. And this cyclic AMP, then it will cause activation of the protein kinase A. So protein kinase A will cause, either it will cause activation or inhibition of the metabolic enzyme by their phosphorylation, or it will cause phosphorylation of the transcriptional factor, so that genetic expression or response is altered, or it will cause phosphorylation of the ion channels, so that ion channels are opened and closed. So in this process, ligand is the first messenger and cyclic AMP is second messenger. So once ligand will bind to the G stimulatory receptor and G stimulatory proteins are activated, the cyclic AMP level of the cell is raised. So once the action is performed, what happens, that adenylyl cyclase, it is inhibited by the G inhibitory. So it has two domains, one is for the G stimulatory and second is for the G inhibitory. And once cyclic AMP has performed its activity, it is inhibited by enzyme phosphodiesterase. What phosphodiesterase will do, it will convert cyclic AMP into AMP. So this is all about the G stimulatory pathway.